Interphase is the longest phase of the cell cycle. If you look at the cell cycle, kind of like a pie, cut up into 24 pieces, 23 of those pieces would be interphase, and one piece would be mitosis. During interphase, the cell does what it normally does. Some cells enter a phase called G0, and this means that they never divide again. They will stay in that phase forever, such as our nerve cells. They're not capable of dividing again. Muscle cells will enter G0, and if they are torn, they will have to repair. They'll have to exit out of G0, get ready to divide, and divide for repair. So normal cells will just go through G1, S, and G2. During G1, the cell will increase in size and the number of organelles will double. Remember, right after mitosis, a cell is small, and it has to increase in size with number of organelles. During the S phase, the DNA is synthesized. So you can think of S as synthesis of DNA. That is, each chromosome is replicated and held together by the centromere. During G2, certain proteins must be made, specifically microtubules, and the centriole must be replicated in order for cell mitosis to take place. So during replication, during the S phase, the DNA, which is a double helix, unwinds, and the enzyme DNA polymerase is going to assemble the new DNA strand. So the old DNA strand is used as a template, and the new bases come in through complementary base pairing, and another enzyme called a ligase is the glue that's going to bond it all together. This process works in one direction, so one strand is replicated a bit differently than the other strand of DNA, but you can see here that as the DNA unwinds, each segment is going to act as a template for a new piece of DNA to be formed, complementary to the old one. And in the end, you're going to have two identical DNA double helixes. Mitosis is the division of the cell nucleus. Following mitosis is division of the cell cytoplasm called cytokinesis. These are not parts of the stage of interphase. They are completely separate. Think of the term PMAT. PMAT tells you the order of the stages of mitosis. P is for prophase. During prophase, the chromatin, which is the stretched out form of DNA, is going to coil and form chromosomes. The replicated centrioles are going to move to opposite poles. The spindle fibers are going to connect the centriole pairs. It's a long phase. During prophase, also the nucleus has to disappear. Remember, the stages are PMAT. Prophase is the first one, metaphase is the second one. Think of metaphase as middle. The chromosomes are in the middle of the cell. That is called the metaphase plate. The third stage is anaphase. Think of anaphase as apart, A for anaphase, A for apart. And here are the chromatids. So a chromosome consists of two chromatids held together at the centromere. Remember those, the DNA was duplicated. And so those chromatids separate at that centromere and become individual chromosomes. The last phase, telophase, is basically the opposite of prophase. The nuclear envelope forms, the chromosomes uncoil to form chromatin, and the spindle apparatus disappears. The very last stage, cytokinesis, is the division of the cytoplasm, and in animal cells with a very soft and flexible cell membrane, we get what's called a cleavage furrow forming between the two cells. This often starts in telophase, continues through telophase, and at the end of cytokinesis, you will have 2 complete new cells. Mitosis is involved in cancer. Cancer is basically cells that are undergoing mitosis too rapidly. So something has happened, a mutation or change in the DNA, that has involved cell replication or mitosis. So cells that divide actively are often cells that become cancerous, such as skin cells and intestinal lining. A tumor is a neoplasm. It's a mass of cells. This is not necessarily dangerous. A benign tumor stays in place, does not move throughout the body, and may only affect you if it blocks something else. For example, a tumor in the ureter might block urine going from the kidney to the bladder. Malignant tumors are more dangerous. Mal means bad in a lot of different languages, so malignant is a bad tumor. And what's happened with a malignant tumor is it has spread. Even one cell can break off and spread to other tissues. When the cell goes to another area, it forms a secondary tumor, but also it releases chemicals that are going to grow blood vessels to that tumor to supply it with nutrients.